Sharing screen. Well, thanks so much for the invitation. I always love to interact with Rotary clubs. I used to be a member of the Ancaster AM Club until my role as Vice President Research prevented me from attending any meetings. But for many years, I was part of that club and just absolutely loved my time in Rotary and hope at some point to be able to get back. So thanks again for the opportunity to tell a little bit about some of the research at McMaster. I come at this with two hats, both as Vice President Research, and I love bragging about all the great research that our colleagues are doing at the university, but then also my own lab is heavily involved in working on SARS-CoV-2. So when we think about how to respond to any sort of infection, there's both vaccines and antivirals. Vaccines are really the proactive, so they protect against an infection. Antivirals are more reactive, they help treat an infection. Currently worldwide, there are about 7 approved COVID vaccines. I'm sure you've heard about most of them in the news. But we don't have any approved COVID-specific antiviral drugs, none that have shown. Some have been tested in clinical trials, but none that have passed through clinical trials. And that is something that both my lab and other labs at McMaster are actively working on. So this, just for your own information, this is a great website, Our World in Data. I have the website down below. And this keeps daily statistics on a whole range of different aspects of COVID-19. So it's a great website if you're interested in statistics and watching the trends. I gave a talk to another Rotary Club about 2 weeks ago, so I just updated this slide from, I think, May 2nd, and even for me, it was amazing to see the difference between looking at all of the data from day 2nd up until just a couple days ago. So it's a really great site that shows, I mean, this particular one is the number of daily vaccine doses per 100 people. You can see that Canada right now is doing remarkably well. And so if we also look at the number of people who have received at least 1 dose of COVID-19, again, about 2 weeks ago when I gave this talk, we were only about halfway in between world statistics in the United States. And within that 2-week period, we've really caught up. And we're almost at the same share of the population as in the States, which is really great to see. And it's great to see all of the rollouts. And I know certainly, I read even in the paper today that it only took half an hour for the new, with the opening up from 18 and up to book all of the available sites within the city. So it's great to see that so many people are getting out and getting vaccinated. So these are the common questions that I get, I would say, almost on a daily basis. One is, how did they make a vaccine so quickly? Were there safety steps missed because it did roll out so quickly? And then, of course, how effective are the vaccines? And so what I would have to say is that, yes, these vaccines were made very, very quickly, but it certainly wasn't by missing any of the normal steps or procedures. In some ways, we were fortunate because of the original SARS pandemic in 2003. There was a lot learned about that particular virus. This virus, even though it's slightly different, structurally, it's very, very similar. So as soon as it was recognized that this was a related SARS coronavirus, we didn't have to figure out what is the virus, what do we make vaccines against. There was a lot of information from the original SARS pandemic in 2003, even to the point where phase 1 clinical trials had rolled out with some vaccines being produced at that point. So we were already several steps ahead. And so that really allowed research groups around the world to mobilize remarkably fast because we knew a lot about the virus. We knew about the spike protein. We knew that it was spike protein that we would want the vaccines generated against. And of course, there's a lot of technology already in play in how to make vaccines, how to produce them. Even with the mRNA vaccines, even though we hadn't yet had mRNA vaccines, that technology has been studied for a number of different types of diseases and other indications. So again, the technology wasn't new. So we were able to mobilize really, really quickly. And even into getting into clinical trials. So all of these vaccines have gone through clinical trials, phase 1, 2, and 3. And when you have such a global pandemic where it truly is global and all countries are affected, it's amazing how quickly things can get done when you have motivated pharma, motivated governments, motivated organizations to really allow approvals to go through and funding to be mobilized. And so it didn't surprise me actually that we were able to get some vaccines out so quickly. No safety steps were missed. We did go through all the full phase 1, 2, and 3 clinical trials. Now, because of the need to get through those clinical trials and the safety to get the vaccines out, not as many questions might have been asked in some of the clinical trials. So this is why, for instance, we knew a lot about the ability of the vaccines to prevent against disease and death, but we didn't know as much about, do they prevent you from getting infected and having an asymptomatic infection? Because that wasn't tested in the clinical trials. That would have been exceedingly challenging and very, very expensive. But certainly from the safety perspective, no steps were missed. And now, of course, we know a lot about the efficacy of the various vaccines, both after your first shot, your second shot. Unfortunately, these are viruses in general, coronaviruses that aren't really immune stimulatory. Even folks that were infected with the original SARS and recovered don't have lifelong immunity against that infection. We've already seen individuals who early in the pandemic became infected with SARS-CoV-2, getting reinfected. So these are likely to be vaccines that will become seasonal vaccines. Now, whether that's every year, every 2 or 3 years, I don't know, but these will likely be vaccines that, become a regular routine seasonal vaccine, similar to the influenza vaccines. So specifically at McMaster, this is a great colleague of mine that works within the same research group that I'm in, Dr. Zhou Zhang. And there's a collection of individuals within our group, the McMaster Immunology Research Center, that have really, really deep strengths in vaccines and immunology. And just a quick fact, 2 of the current vaccines are based on adenovirus vectors. And that technology of using adenovirus vectors for vaccine production was actually first discovered at McMaster many, many years ago from Frank Graham. So that whole technology of using viral vectors and using adenovirus vectors is a McMaster-based discovery from probably 30, 40 years ago. And so what Zoe and others are working on, and we're helping not directly, but a bit more indirectly because we're working with the virus in the level 3 facility, is a development of what we call a second generation vaccine. So unlike the current vaccines, all of the current vaccines are directed just against spike protein. And spike protein is that protein on the outside of the virus. So it's what's sort of available for the immune system to see. Instead, this strategy is against spike, but 2 other proteins of the virus that are more conserved between not just the original SARS and SARS-CoV-2, but also conserved within the coronavirus family. And so by making this second generation vaccine that is really generated antibodies and protection against 3 different types of SARS proteins, we're really hoping, and we're just about to launch into some phase 1 clinical trials, really hope that not only will we get antibodies and protection directed specifically against spike, but also against other proteins. And so then your immune response will be much, much broader. And so not only broader against potential variants, because a lot of the variations and a lot of the mutations we're seeing are specifically in spike. So not just a broader immune response against variants, but also against other coronaviruses. There's already 4 known human coronaviruses that cause common colds and common upper respiratory tract infections. There's likely to be more pandemic coronaviruses. We've had 2. I'm sure we're going to have a 3rd and a 4th and a 5th, hopefully not for a very long time, but the possibility is there. And so this strategy of building a broader type of immune response will hopefully help protect against other coronaviruses as they come up. This is also similar to almost all except for the Johnson & Johnson, a 2-dose vaccine. But one of the differences is that the second dose is really to be delivered into the respiratory tract. So it's an inhaled vaccine. And Zoe and his colleagues have done a lot of work with vaccines against tuberculosis and have really shown that by delivering the vaccine directly to the site of infection, in this case into the lung, you induce a much, much better immune response, and it's more directed against to where that infection is. The other advantage to having an inhaled vaccine is you need about 100-fold less in the dose. And so when you think about manufacturing, and certainly vaccine manufacturing has become a huge issue globally, but also in Canada, when all of a sudden now you need 100-fold less for every dose for the inhaled vaccine, you can see how that can be really advantageous. A lot of these initial vectors for a lot of the preclinical trials were actually produced in-house. So we have a vector laboratory, a GMP laboratory. It's not big enough to make the types of millions of doses for phase 3 clinical trials, but certainly for some of the preliminary work. And this is a vector facility that has been in operation for about 17 years. So it really positions us quite well to do some of the initial studies, and certainly with all of the funding from the federal government on biomanufacturing, we're really looking to become part of that whole pipeline in the manufacturing space. So some of the other COVID related research at McMaster, I could probably spend the next 3 hours talking about this. There's a lot of work going on. During the pandemic, we were very lucky to be able to keep some of the research going on campus, those that had to access infrastructure. And so, we had a lot of work going on over the last year. We didn't have to completely shut down, which was really great. We have a lot of development and testing, as I mentioned, of new drugs and antiviral therapies, something my lab directly is working on, but in collaboration with others. We are one of the few universities across the country that have a containment level 3 laboratory, so we can work safely with SARS-CoV-2. In-house, and the virus that we and everybody else across the country is working on is one of the viruses that a postdoctoral fellow of mine was able to harvest from patient samples in Toronto well over a year ago now, last February. We have a lot of expertise that's doing, and again, looped into both provincial and federal initiatives to sequence all of the clinical isolates and to track mutations. So we have a large group involved in sequencing and tracking mutations. We're doing a lot of work actually with wastewater and wastewater testing, again, a provincial initiative to be able to track virus within wastewater. It's been proven to be very, very effective, especially, for example, for McMaster, testing wastewater coming out of residences. This will be part of our safe reopening plan for the fall. Windsor, where they've had people on campus over the last several months, have shown that by testing wastewater, they can see that they have the beginnings of an infection before it becomes apparent actually in the students. And so they've been able to use that to then go into residences, screen all of the students, and find those that are asymptomatic. So we have a lot of work being done with wastewater testing, development of new diagnostics, using, instead of the nasal pharyngeal swabs, using saliva, so a spit test, a number of paper-based tests, so it's almost like a pregnancy test. So these would be low cost, at home type of tests. They're just being validated now. We have some really good data that they can also pick up on a lot of the variants. So those are just getting clinically validated. A number of different clinical trials coming out of McMaster, not just antiviral drugs, but a very large trial with convalescent serum. So that's using the serum from patients that have recovered from COVID and using the antibodies that are naturally generated to help protect those that are in critical condition. A lot of work from our engineering faculty on testing PPE, for example, working with companies, both local companies that have sterilization methods. We did open and had received funding from the provincial government, a center of excellence in protective equipment and materials. So not just testing PPE, but actually developing new materials for enhanced PPE production. And then we have many, many different projects on the social sciences, looking at the vulnerable populations, looking at community-based projects, business-based projects. So it's been a really, really busy time at McMaster. Another project that I've been actively involved in, this is a great collaboration between McMaster and St. Joe's Research Institute. It's McMaster Health Labs. It's a nonprofit organization that has really been focusing on curbing that human, economic, and social impacts of COVID. And it's really through a research-based initiative to help companies enable safe return to work. And so it's allowing companies to make really evidence-based decisions on how often they should be testing, what type of testing, so that they can really open their businesses and do it in a safe manner. The first study we did was way back in the fall. It seems a long time ago. It was back in the fall with Air Canada. This is when we were helping to screen all incoming or the ability to screen incoming international travelers. And so that really gave the first indication of even going through the logistics and operationalizing, and also gave some information on percentages of travelers that were coming in, how many were shown to be positive even if asymptomatic when they landed versus within the next 2 weeks. And so now we're working with Shelter Health Network, for example, in the city of Hamilton. So working within the shelters to do a lot of testing, but also working with a number of other companies to really enable that asymptomatic testing. So that's a project that we're very, very proud to be a part of from McMaster. And then finally, probably the biggest project that we're working on at McMaster is the global nexus for pandemics and biological threats. So in many ways, when the pandemic hit, this was sort of within the sweet spot for McMaster. We have very, very deep strengths in health sciences, in all aspects of health sciences. We've had for 15 years a world-renowned Institute of Infectious Disease Research. And so that institute that worked on infectious diseases, antimicrobial resistance, but also, McMaster is really well known, and that's what I love about working at McMaster, especially in the research portfolio, is that true multidisciplinary and evidence-based way of looking at big problems. And so this global nexus for pandemic and biological threats, we're really bringing together it's not just a McMaster initiative, and so it's truly a global initiative across Canada. We have really good connections with a number of institutions across Canada, with large pharmaceutical companies, with governments, with policymakers, and it's really bringing together all of those players, so that to solve a really complex problem like a pandemic, you need complex solutions. So if you are, for example, thinking of a vaccine, it's not good enough just to make the vaccine. You need to think about supply chain. You need to think about the cost. You need to think about vaccine hesitancy. You need to think about all of the, the social and the political issues that surround vaccines. You need to think about how do you distribute vaccines in an ethical manner. So it's not just about the science behind a vaccine, it's all of these other issues. And that's just thinking about vaccines. And so we've been really working on, bringing together, to really think about, how can we as a university have impact locally for the city, but also have impact nationally and globally. And so, there's been a lot of work on what the activity would be, but there's also work on where are we going to house this initiative? It's really a hub and spoke model. And what better place to house this than at the McMaster Innovation Park? So as you're probably all aware of where the McMaster Innovation Park is, here's just a schematic. You can see the 403 up on the upper left. And so this building down in the bottom right, this old, we call it the glass warehouse. It's a little bit of a fixer-upper. It was actually used in the war to make tanks and artillery. That's part of what we have at the Innovation Park. And what we really want to do is turn that glass warehouse into this. And so this would be a hub. It would be the center focal point of the Innovation Park. And it's beautifully situated for these activities because it's not just about research, it's also about then enabling those findings to really go out into the hands of the business community, the community-based community members that can really enable the work that we find to disseminate out into the community and into industry or commercialization. And so housing this at the Innovation Park is going to be incredibly important. And so that's where I've been spending a lot of my time in fundraising and growing this initiatives, which we're really, really excited about. So finally, thanks so much for the invitation, and I will stop screen sharing and happy to answer any questions.
Wonderful. Thank you. Questions, comments? Janet McNaught and then Paul Lake.
Oh, I still stop share. There we go.
Janet, go ahead. You are muted.
I was trying to find my unmute. Karen, in the early days of pandemic tracking, I know they said that the virus was slow to mutate and the range of mutation was fairly small. So there were 1 strain and 2 and 3 and 4, but they were all fairly close in their characteristics. So they would be considered to be 1. Has the rate of mutation increased over time? Are they more difficult to sort of treat as 1, or is it still within a range that's stable?
So all viruses mutate, that's a normal, and all viruses have a different mutation rate depending on, the nature of the virus. So the mutation rate of the virus per se has not increased at all, because it's all about the polymerase and about, the actual replication of any particular virus. But when you take a mutation rate times the number of viruses that are out there globally, it becomes it's a math issue, right? So, and it's a probability issue. So the more the virus has spread globally, even with a relatively low mutation rate, the number of those instances, obviously, as the virus has grown to be really global, we see more and more mutations and different types of mutations. I would say probably 99.9% of the naturally occurring mutations have no advantageous or are not advantageous at all to the virus. So we never see them. It's only those really small number of mutations that provide an advantage to the virus and a selective advantage to the virus that will then propagate out. And so and this is where you hear about it being a race between the mutations and the variants and the vaccines. The more we can get people vaccinated and, we decrease the number of viruses or the number of people that are infected, then again, that probability of generating a new variant goes down.
Right, because each
Wonderful,
virus
thank
is
you.
a host.
I'm going to go to Paul Lakin, then Cheryl, then John Janice. Paul?
Yeah, Karen, I'm an alumni of McMaster, and this is what the work you're doing makes me very proud. It's really excellent stuff that's going on. So yesterday there was an announcement by the federal government about a new vaccine manufacturing company in Ontario. Would McMaster play a pivotal role in that, the development of the vaccines there?
Yeah, so we have we're building partnerships with just about everyone that's in this space, and so when you think about vaccine production, the work it's a huge continuum all the way from that really early stage research and discovery through to what we can do in-house in our vector facility, which is we can pivot very, very quickly and do a number of different types of vaccine production because we're smaller scale. So for preclinical, and then, once you get beyond that, then you can start to work with some of these other, manufacturing capacities where they have much, much larger capacity, but they don't have the ability to be very nimble. Because once you get to the really large vaccine manufacturing, and they're also building one in Quebec, in Montreal, like from the National Research Council, which will be making tens of millions of doses at a time. They don't have the ability to just switch production and be really nimble to try out something new. So right now where we sit, it's at sort of the beginning of that full continuum, but it's critically important for us to partner with these larger manufacturing capacities, but they also are willing to partner with us because we can do that early R&D. And so it does become we think of it as an entire ecosystem. And that's what we've been really aiming to build, not just at McMaster, but at the innovation part, is that ecosystem.
Thanks, Paul. Cheryl, and then John Janice.
Yes, my question is about trials. For the AstraZeneca, I received my first dose, and it said they were waiting for trials before the second dose. Is there any way Canadians can volunteer? Because it keeps saying they're waiting for the UK studies. So are they testing any clinical trials for Canadians to see about the second dose of AstraZeneca?
Not that I'm aware of. I'm not saying no, but not that I'm aware of. So the AstraZeneca was an initiative out of Oxford University. All of the, a lot of the manufacturing, the major manufacturing is in the UK. And so that's where they're doing a lot of the, the clinical trials and the clinical testing. I haven't heard of any, doesn't mean there aren't any here, but because all of the activities and the manufacturing was based initially from Oxford, but in the UK, that's where they're doing the majority of their studies. And I think because that's where they did a lot of their phase 1 studies, they also have all of the infrastructure in place, like the enrollment and the reporting, and that's all in place there.
Thank you.
Thanks, Cheryl. John Janice.
Hi, thanks. You mentioned about the race between the variants and the vaccines. I suppose that presumes that the vaccines stop the spread, but is there actual evidence, is there growing evidence of that one way or another?
Yeah, so they are seeing in, in areas where they have really good vaccine uptake and where the, areas where they're now approaching, there are some areas, for example, in the States where they're approaching, 70% of the population, being vaccinated, either partially or fully vaccinated. And it's not clear what is the number, what's the percentage that you need for herd immunity, but it's certainly, once you start approaching 70, 80%, you start to see evidence of herd immunity where now incident rates are going down. And numbers of individuals being infected are decreasing. Of course, that's going to depend, the efficiency of that happening is going to depend, are you in an area where, you know, you have a very fast spreading variant, or you have the original? I mean, it's hard to say absolutes, but certainly, and I think even, you know, somewhat locally, and within Canada in different regions, we're seeing where we do have that vaccine uptake, numbers are slowly starting to go down.
Wonderful. John, thank you. Dr. Mossman, we're coming up against 8:30, so we need to come to a close. Thank you so much for spending time with us today and on a topic that we've all been paying a lot of attention to. We don't send, we don't provide a thank you gift. We are going to send sufficient funds to inoculate 25 people as part of the Rotary International Polio Eradication Program on your behalf.
Oh, fabulous. Thank you.
Fits in perfectly.
Fits in perfectly.
Thank you very much. And
Yeah,
folks,
well
we're
done.
coming to an end. Round of applause, absolutely, for Dr. Mossman. We next week have a club assembly, and John Dalgleish will be taking us through the goals for the next year. So looking for some input and for some commentary on those, and let us all finish with a toast to Rotary International.
Karen, before we leave, Karen and Mr. President, could I ask you just to stay for a quick moment after?
Sure.
Thank you.
Thanks all. We'll see you next week.